Hi, my name is Cara. I'm a master's student here at OSU. Today I will be demonstrating genome size estimation of butterfly bush cultivars using a flow cytometer. The first order of business is collecting good fresh plant material. The most recently expanded leaf is usually a good choice. We need to collect plant material of our butterfly bush samples and our internal standard. I will describe what an internal standard is later on in the demonstration. Here in the Ornamental Breeding Lab, we use flow cytometry to complement our breeding program. Ploidy manipulation is an important piece of our program and is versatile from creating reduced invasive potential cultivars to generating novel traits. It is not easy to measure by conventional means, and the flow cytometer saves us many hours on the microscope counting chromosomes. The flow cytometer allows us to estimate genome size via relative fluorescent activity. This means plant DNA must be extracted and stained with a fluorophore tag. Today we will be using special kits from Sysmex designed for use with DAPI. DAPI is a non-DNA intercalating dye, meaning it won't accidentally bind to your DNA or RNA. It is quicker and safer to use than alternative dyes such as propydium iodide. To run our samples of butterfly bush and internal P standard, we're going to need cleaning fluid, fresh sheath fluid, filters, Rarin tubes, nuclei extraction buffer, staining buffer with DAPI, petri dishes, a double-sided razor blade, plant samples, and internal standard plant. The internal standard is a plant that has a quantified genome size and is easy to run through the flow cytometer. The flow cytometer measures the relative fluorescence of samples, which depends on how well the dye binds to our nuclear genome. This can vary from sample to sample and preparation to preparation. We run plants of interest and the internal standard in the same preparation to mitigate this issue. The genome size of the internal standard is used to estimate genome size of the sample plant along with their relative fluorescent levels. Today we are using P as the internal standard. The measured genome size of this cultivar, Cytrad, is 8.76 picograms. I am selecting leaf tissue parts and fresh leaves well away from the midrib or prominent plant veins. This is to avoid including secondary metabolites in my sample, which can interfere with dye binding. I need to collect about 1 square centimeter of each sample. I'm collecting one sample with just internal standard and one sample each with my study plant and internal standard together. We are measuring the genome size of 2 butterfly bush cultivars. Buddleia exwayeriana honeycomb is an interspecific hybrid between species of different ploidy levels. It is a cross between tetraploid Buddleia davidii and South American diploid Buddleia globosa. Black Knight, our other cultivar, is a tetraploid Buddleia davidii cultivar. Before we can stain the nuclear genome with fluorophore dye, we need to release whole nuclei from plant tissue. This is accomplished by chopping leaf tissue with a double razor blade in a nuclei extraction buffer. The double razor blade chops finely rather than crushing the delicate nuclei. Over-chopping the sample might interfere with dye binding, so you'll want your sample to look something like this. Next, the buffer and chopped sample are poured through a filter. This allows nuclei and extraction buffer to pass through the tube, but prevents larger pieces of plant tissue from passing. The filter is tossed into a bucket of lab tone to save for future use. Finally, a staining buffer with DAPI dye is applied to the extracted nuclei. This attaches the fluorophore dye to the nuclear genome for the flow cytometer. I'll speedily prepare the next 2 samples. We are ready to begin running samples to the machine. Prepare the machine with a cleaning solution first. Then, slide the internal standard preparation onto the intake port and click into place to run. The machine begins the run by taking up a pre-specified volume of sample preparation. Next, the machine will line extracted, filtered, and stained nuclei up in a single-file line as they pass through a light excitation phase. At this stage, a UV light source is directed at a single nucleus, and a measurement of the ensuing fluorescent activity is recorded for that nucleus. This is reflected in the histogram on the screen. A running count of how many nuclei the machine has analyzed thus far in the run is recorded in the bottom right corner of the screen. The y-axis of the screen displays the number of nuclei counted. The x-axis of the histogram records the relative fluorescent activity each nucleus produces. The peaks of the histogram may then be gated to take a mean fluorescent activity per nucleus captured in the gate bounds. The first sample contained the P standard only, which is the peak at about 400 on the x-axis. The small peak at 800 are cells in the G2 phase of mitosis that are preparing to divide and so have double the amount of normal nuclear DNA. This is aptly called the G2 peak. I will speed run the next two samples with our plants of interest and internal standard. Notice how the P peak, or the second peak, shifts from sample to sample. This reinforces the importance of running the internal standard together with our plant of interest, as relative fluorescent activity varies from preparation to preparation. I will gate and record mean and CV percentage, or an estimate of error, for both peaks in the histogram. I will show how to use these numbers to estimate genome size of the plant of interest next. To calculate relative genome size, we will divide the mean fluorescent activity of the plant of interest by the mean fluorescent activity of the internal standard. We multiply that by the 2C genome size of the internal standard in picograms. As you can see, the interspecific hybrid honeycomb has a substantially larger genome size than Buddleia davidii black knight. Chromosome counts will be necessary to confirm this is due to ploidy variation, and not due to a difference in chromosome size between the different species.